Hello students, welcome to Biosastra Talks, and we'll move on with the part B from the FAQs from genetics session. Okay, let's get into the topic. So what was the question that was given here is, it is also from the replication itself. So what was the question is, who proved the semi-conservative replication of DNA in E. coli? So in the previous video we learned about what is a replication and what were the enzymes involved in it. Here they are asking about which scientists invented this semi-conservative replication. We know that, right? In the E. coli, obviously you can see the semi-conservative replication in which some strands of DNA are not conserved properly. So this was normally proposed by, what was the correct answer is, with Michelson and Stahl. Okay, so these are the scientists who proposed the semi-conservative replication of DNA in E. coli. So Watson and Crick, we know that double helical DNA and everything, Hershey and Chase experiments. So like that, the Michelson and Stahl are the scientists who proved that semi-conservative replication occurs in the DNA of E. coli. Okay, so we will move on to the next question. What was the next question is, obviously after the replication we'll move on to the transcription, right? So what is a transcription? The formation of RNA from the DNA bonds, like means the formation of RNA molecules from the DNA itself. Okay, so this is the transcription process here. So if we see the question, what was the question they have given is, transcription in eukaryotes require. Normally means we know the transcription occurs in eukaryotes and prokaryotes. So whereas in the eukaryotes, normally what is the main thing that is required for the transcription? Here they have given the options: DNA polymerase, RNA polymerase 2, ribosomes and DNA ligase. So these are the four enzymes that were given here. So DNA ligase is not required for the, so if we move on from the some of the options here, DNA, if we see the options, DNA polymerase is the enzyme which synthesizes the DNA molecules. Okay, so this is not the one. Why? Because here we are producing the RNA polymerase, whereas the RNA polymerase 2 is the enzyme. There are two types of RNA, like RNA polymerase 1 and 2, in which one of them is present in eukaryote and prokaryote. We'll see this is right or not. Okay, if we move on to the ribosomes is not involved in the transcription process, right? Why? Because the RNA is not produced in the ribosome, so this is also wrong. And DNA ligase, what is the role of DNA ligase? To attach the Okazaki fragments, right? So this is also wrong answer. Okay, like the fragments of the DNA, to combine the fragments of the DNA from the lagging strand, the DNA ligase is used. So in these four options, what was the best option we can consider is nothing but RNA polymerase 2. Okay, so here what is the best option is RNA polymerase 2 is the enzyme which is responsible for the transcription in eukaryotes. If you see the picture here, so what is the transcription and everything? Transcription means preparation of the RNA, no? So here you can see that one coding strand and one template strand. So here you can see that synthesis of the RNA from the template strand itself, that is nothing but RNA polymerase. If you see the in a sequence manner, this is the coding strand and this is the template strand, and the RNA is produced from the template strand itself, means the nucleotides or nitrogen bases that are present in the template strand will code for the the nitrogen base pairs will combine, will be formed from the nitrogen bases on the template strand of RNA. Okay, another thing you have to remember here is, means template strand is the strand which is responsible for the synthesis of RNA with the help of RNA polymerase. Okay, so and RNA polymerase is of two types, polymerase 1 and 2. And another thing is a coding strand and template strand, they are having the primes 5' to 3' and 3' to 5' directions, like carbon thing. 5' and 3' means carbon molecules, okay, positions. So if you see that, so another thing you have to remember is from where to where, normally they'll ask that from where to where the translation will takes place, means from 5' to 3' or 3' to 5'. If they asked about the template strand, you have to say that it is from 5' to 3', whereas the RNA synthesizes in the form of 3' to 5'. Okay, means in the reverse process. Like that means according to the DNA strand, the RNA is synthesized in a reverse process. Okay, so like that it will happen. So this is total translation process, and in which I told you know that the RNA polymerase is the right answer. RNA polymerase 1 is responsible for another thing, whereas RNA polymerase 2 is the main enzyme that is important for the transcription. Okay, we'll move on to the next question here. So obviously, what is the next question? From the translation itself. So what is the function of tRNA during translation, they are asking here. So if you know the translation means what? Synthesis of proteins, okay, or the amino acid chains from the what? Yeah, from the mRNA, or like a mRNA, right? Simply you can say that synthesis of proteins from the mRNA with the help of the ribosomes. Okay, so that is nothing but the RNA, sorry, translation here. If you see, this is all the translation unit in which you can see that the mRNA synthesized from the DNA through the transcription enters the ribosomal unit. In between the larger unit and smaller unit it is placed, and it will move on like this. Where it is placed, obviously according to the sequence on the mRNA, the opposite genetic code, means the opposite amino acids, the codons, are attached in the ribosomal, larger ribosomal unit of the ribosome. So those are nothing but anticodons, means the codons which were attached against the codons of the mRNA are known as anticodons. So according to the sequence, anticodons are attached to the mRNA, and like that the proteins will be formed. What is the protein? It is a sequence of genetic codes, no, like 2. So like that it will happen, and again another anticodon will come. It will move on, means 3 codons will combine to form one amino acid. So like that the amino acids will form, the amino acid peptide chain is formed to form a protein. Okay, so like this it will happen. There are 3 types of units in the ribosomal unit and one smaller ribosomal unit. So this process whole takes place with the help of the mRNA, tRNA, ribosomal units, and these anticodons and codons. Okay, so here what they are asking, what is the function of tRNA, they are asking. So here in the picture you can see what is the tRNA doing, the transfer RNA. It is transferring these codons or anticodons to the what? To the, or away from the? Away from the, you have to say that, away from the or to the, actually not away from the. Why? Because it have to like what they'll say, it have to bring down those codons, okay, codons from the remaining cell environment into the ribosomal unit. So that means according to the mRNA sequence, the anticodons are present. So the anticodons which were coding for certain amino acids will form a chain like this, and it will go on like this to form a protein. Okay, so according to that, according to the mRNA sequence, obviously the type of proteins are defined. So that tRNA function is, it carries the codons to the, like it transfers, simply you can say that it transfers or carries the amino acids to the ribosome. Okay, not from the ribosome, to the ribosome. So this is the right answer. Here you can see the options here. First one is, it forms the peptide bond. No, tRNA does not help in the formation of peptide bond. Another thing, it carries the amino acids to the ribosome. Okay, you can consider this option. And third one is, carries codons. It will not carry codons, it carries the amino acids itself. And fourth one is, it joins mRNA to ribosome. No, it will not join the mRNA, like it only helps for the transfer of anticodons. Okay, so what is the right answer here? You can say that the transfer RNA carries the amino acids to the ribosome, not away. You have to say, see this, it's not away, it is to the ribosome. So like this the translation will take place. If we move on to the next question, here they have asked about the operon model of the gene regulation, as we know. So here they are also asking about the scientist who is helpful for the operon model. Okay, here also answer is here. Actually, who proposed the operon model of the gene is nothing but Jacob and Monod, and you have to think. So these two are the scientists who have proposed this operon model of the gene regulation, means the gene will regulate to show certain functions. Okay, so if we move on to the next question, here this question is came from the like laws, Mendelian laws, or like dominance, codominance, etc. So here this question came from the incomplete dominance. So in incomplete dominance, the F1 phenotype is, means here they are asking, in the incomplete type of dominance, what is the F1 generation phenotype? Okay, how it will appear and how it will look here they are asking. So for that, if we see the table, you can understand easily. Here you can see that the spectrum of dominance. Normally dominance is of three types. One is complete dominance, another one is incomplete dominance, and third one is codominance actually. So codominance means the characters will appear both from the parents, means equal priorities. Okay, so whereas if we see the complete and incomplete dominance, here complete dominance means the F1 generation will get any one of the character from any one parent, not from both the parents. Okay, so whereas in the incomplete dominance, if you see the two parents which were having two genotypes, okay, so genotypes and phenotypes, uh, in the F1 generation you can see the mixture of these two parents, means the offspring will get the characters from both the parents, and the offspring is different from the parents, so that is known as incomplete. Why? Because any one of the parent is not completely dominant on the other, that's why it is incomplete. Means both of them are equal in participation, that's why they'll show the equal participation to give a new type of phenotype here. Okay, so these are the F1 types of generations. If we move on to the F2 generations, you can see that whereas in the complete dominance you can get majority 3:1 ratio, whereas is the in the incomplete dominance you can see that 1:2:1 ratio, like the dihybrid cross. Okay, so like this it will happen. So if we move on to the question, what they are asking in incomplete dominance, what is the F1 phenotype? So F1 phenotype, what is it? First option is same as the dominant parent. So if we see the incomplete dominance, the F1 phenotype is same as like dominant parent. No. Okay, it's not right. If we, another option is same as recessive parent. If you see the recessive parent white color flower, it is white color in white color, so it's not white in color. So this is also wrong. Mixture of both. If you see both, yeah, this phenotype is having F1 phenotype is having both the parental characters. That's why it is a mixture of both the parents. Okay. So if we move on to the next option, it is completely different. It's not. Is it completely different? Means completely different means you can say it in another way, means it looks like completely different, but if we think it in a genetical way, it contains both the parental genes, right? So it is not completely different, means it is having both of their genes. So you can say the answer as a mixture of both. Okay, so option C is the right answer here. Okay, so if we move on to the next question, what is the next question here? Yeah, very good. It also came from the dominance itself. What was the question they are asking? The allele that makes that masks the expression of another, means the allele or the character that marks the expression of another allele expression is depressed or suppressed by another allele. So that is known as what? Okay, what is that? Is they are giving the options, that is known as recessive allele. Recessive allele cannot mask the expression of the another. Why? Because it is not that much active. Okay, and codominance means it will you can say that it will what will say, it will not mask, it will combine with the another one, it will not mask the character of the other. Okay, what it will do, it will combine with the another allele to give a different one. Okay, so the codominance is also wrong. What it is not the right answer for this. And if we go on to the dominant molecule, dominance means you can see that obviously dominancy means one person is dominant over the other, means he is masking or he is suppressing the other one's actions. So like that only here the dominant genes will mask the expression of the another. If you see the complete dominance here, dominance means here the purple color flower has completely dominated these white color flowers, and the F1 generation got only purple color, right? So it is completely dominating it. So this is the dominant allele. The allele that masks the expression of the other is known as the dominant allele. So okay, this is the right answer. It's not complementary. Okay, 1 minute. Next one. If we move on to the next question here. So what is the next question is, industrial melanism is an example of, we know that it is a type of melanism, means the skin color that occurs in which type they are asking. According to this, what is the example in industrial melanism? Melanism means what? Industrial melanism means the people are getting darker and darker due to the industrial pollutions or like the dust that came from the industries. Melanism means the darking, melanins or the pigments that were responsible for the skin color, right? So industrial melanism means the darkness of the people over the ages or over the evolution due to industries and everything. So this is an example of what they are asking. Is it the mutation? No. If the mutation occurs means it will suddenly occur. So this is not the industrial melanism. Why? Because industrial melanism occurs by step, means like over the evolution. Okay, this is not the right answer. Whereas the genetic drift is also not. Why? Because it is not genetically occurred. Okay, and another one, natural selection, and fourth one is gene flow. Gene flow means, one thing you have to remember, gene flow means flow of genes from one parent to the another, like that. If in the parent one he is having a lesser like fair color, but the parent like next generation is not fair in color, it's not completely dependent upon the genes. It also due to the industrial melanism who are living in the industrial areas. So in these four options you can say that the natural selection. Why? Because naturally they are selected because of the nature. So that's why industrial melanism is the example of natural selection. Okay, if we move on to the next question, they have asked about here, what is that? If a gene, principles of inheritance. Okay, if a gene is located on the X chromosome, it is called. So here they are asking, if a gene is located on the X chromosome, what it is called. Here you can see I have given one chart here, like how you can see that if a gene is located on the X chromosome, what you can say according to the laws of inheritance. Normally you can say it, first one they are saying it as autosomes. No, it is not an autosome. Okay, autosomes are these ones, not the genes. Okay, the genes are not autosomes, so these are the autosomes. And Hollandric genes, obviously they are not Hollandric genes. And sex linked genes. Why? Because sex linked genes, why because they'll come from through the what they'll say, contact of the parents. Okay, so another thing is sex linked genes. And codominant gene is not present on the X chromosome. Okay, so if we see the sex option here, the option is sex linked gene here. So if we see the flow chart and everything, I'll explain you what is a sex linked inheritance and everything. If you see that the parent who is first of all the unaffected father is there, like with any disease or something. If you see hemophilia, H means hemophilia, he is an unaffected father, he is having the hemophilic gene on the X chromosome. Okay, when he is having the kids, okay, when like if you see that he is unaffected, no, he is having two zygotes, and in that zygotes normally, if you see that XH and XY means XH and Y, so these are the two zygotes present in the like what they'll say unaffected father. Okay, XH which is having the hemophilic gene and Y gene, and another side we are having the carrier mother who is having she is not hemophilic, but she is carrying the hemophilic genes which are not dominant in her, means they are recessive, they are suppressed in her, means she is a carrier one. So the carrier mother, she is having one dominant gene and one recessive gene. When these mother and father were contacted each other sexually, what will happen is we'll get the result of these parents. Like if you see, one kid can have the chance of having the hemophilia and another cannot be affected because of the Y chromosomes, and another kid is like she is a carrier. If she is having both X chromosomes, she is a carrier, and the last one is also having the what they'll have hemophilia. Okay, like this. Okay, this is the here the small h is nothing but the hemophilia here. So here half of the sons are having the hemophilia. So if you see that in these four kids, one daughter is not affected with the hemophilia, and half of the sons are not affected with the hemophilia, and maximum daughters are not affected. Why? Because here the mother, the genes which were carried from the father and mother will not, if in this case, if the father and mother both of them are hemophilic, then you can say that daughters can also be affected. But here in this case only half of the sons will have the hemophilia, because all of this is because due to the sex linked gene that is present on the X chromosome. Okay, so obviously you can say that maximum carriers or mothers, it will be not by the fathers, but it's in some cases it will take place, but maximum carriers or mothers. When these carrier mothers are infected, like when they contact with each other, they'll get these type of results in which only half of the sons will have hemophilia. So this is the hemophilic chromosome. Okay, if we move on to the next question, what is the next question is, which one of the following is an example of polygenic inheritance? They are asking. Polygenic, the name itself can say that. What is it? Polygenic inheritance means, poly means so many genes. Okay, inheritance means not only one gene, so many genes will come. Okay, what is its example? If you see that they have given the 4 options here, and you can see the picture also, how you can say what is a polygenetic inheritance. If you see that there are 2 types of parents who are having polygenes, which were responsible for any one character, and they are carrying for polygenes, like one is for color, one is for size, or like that. So when these 2 parents were crossed, what was the result is the phenotypes are also having both the genes, like some of them will have dominant and some of them will have recessive. So like that it will happen. And then when you see the result here, you can see that they'll have, I mean, some of them will have some of the like characters from both the parents, and some of them will have characters from both the parents. So like this will happen. This is known as polygenic. Not only one character, you can say that this person is having height, less weight, and fair color, and this person is having less weight, means dwarf, and black in color, and like that. So means each single kid or offspring can get more genetic variations or more genes from both the parents. It is known as polygenic inheritance. Okay, here you can see the options how they have given, and how many genotypes came or were seen in the offsprings of these polygenic parents. Okay, so here they are asking about the example of the polygenic parents, polygenic inheritance. So what were the options given here? Skin color in the humans, blood group. Can you say that the blood group is the right answer? Why? Because blood group is also, normally if we think that blood group is also due to different genes, right? Okay, another thing, tongue rolling. No, no, no. Color blindness. Color blindness is also due to not normally due to color polygenic inheritance. It comes from any one gene. And blood group is also one gene only. Okay, whereas not gene like saying character. Whereas in the skin color in humans is not dependent upon anyone. For example, if you see that he is white in color, and she is like a little bit fair. She's like little bit fair, and the kids may be having the medium color, like they are not fair, but somewhat, like some of them will be black, or some of them will be white, and some of them will be little fair. So like that it will happen in the polygenic inheritance. Okay, so what is the best option for this polygenic inheritance is skin color in the humans. Okay, and we'll move on to the last question. So what is the last question here is, which condition is not sex linked? Here they are asking which is not a sex linked condition in the humans. Okay, here you can see that hemophilia. Here only we have learned that hemophilia is the sex linked one. In the sex linked inheritance we learned it. So this is also, it is not the correct answer. Color blindness. Color blindness is also sometimes sex linked. Why? Because the color blindness genes are present on the X chromosome. Thalassemia and Duchenne muscular dystrophy. Muscular dystrophy is not due to sex linked. It comes from like due to the genetic evolution, like mutations or drips like that. So this is not wrong. So option is thalassemia. So thalassemia is the correct answer, which is not a sex linked chromos, like sex linked inheritance. Okay, so like this, these are all the questions that were given from the frequently asked and the PYQs from the genetics chapter itself. Okay. That's it, guys. These are all the questions from the genetics chapter. If we move on to the next thing, and in the another video I'm going to upload some of the FAQs from the ecology chapter. So we know that ecology is also one of the important chapter for the NEET exam. Okay, so what were the questions? I have given some examples. What were the questions included in the next video? I hope all of you are waiting for this video, and if you are interested in that, and just check on to the my next video. And another thing, for today's session, if you like this type of explanation and everything, and if you have any doubts, just comment me in the comment section.